Now, what can I do? How do I share my stream? Authorize one social media account. All right. Not that it matters, but I guess so. What to post to Facebook timeline? Not now. Well, that's the whole purpose of doing that. I turned it off. Okay. Well, it's time now, so I think I'm going to be able to share stream that. Oh, this is exciting. Yay. Okay, so let me see where I'm at. I'm at my. What am I doing? Oh, I want to see if this can. If I can see that this live stream has started. Yes. Okay. It has. So thank you, the three of you that have joined me so far. Oh, let's do this. Okay. And I guess I want to go back to this page. Okay, so it is 9:31 Central Standard Time, though I am in the Eastern Time Zone. That's all good. So we are going through Chapter 10, the biochemistry of the genome, the structure and function of DNA. And I think I stopped a couple of slides in. The learning objectives include describing the biochemical structure of deoxyribonucleotides, identify the base pairs used in the synthesis of deoxyribonucleotides, and explain why the double helix is described as anti-parallel. Okay, so remember we had our base with our deoxyribose sugar and our phosphate, and that the difference between the DNA, the deoxyribose, versus the ribose came in at the 2' carbon on the sugar backbone. And I do know, I put it in the announcement, but I do know that you can see the green screen back here. I'll have to work with this software some more because our UJA and our canvas did not, it crashed out on me. That happens, right? Okay, so next. So recall we had our pyrimidines, we had the Ys with the cytosine, the thymine, which is uracil and RNA. These are the pyrimidines. And then we had pure as gold was brought to us as a mnemonic for remembering our purines, our adenines, and guanines. Okay. Oops, I can't. There we go. So they form linkages from the 3' to the 5' as moderated through the phosphodiester bond. This is another type of dehydration synthesis, which is a very common theme in biological reactions. So this requires DNTPs. Oops. There we go. And it releases pyrophosphates, and they are hydrolyzed to drive polymerization. There's a really fun video from the DNA Learning Center that I expect to show you after fall break, because there's a different chapter that goes through the biomechanics of these processes, and I expect we'll go through that relatively quickly. Okay, let me check in over here. Hopefully the sound is working as it was when I tested it. Oops, I'm sorry. Okay. So again, 3', 5'. We can only add this reaction can only take place whereas there's a free 3' end to attach to. All right. So next. So the race for the double helix should be available to download. I put it into a zip file, which I don't think really compressed it, but it won't try to convince you to watch it through whatever else. You can just download it from the files directory and watch it that way. And I think it's useful to watch. It is kind of slow, but there it is. Because they build this model at the very end. It's like the last shot. Spoiler alert. they did discover the structure of. Okay. So Watson and Crick proposed the double helix model for DNA, and we have a major groove and a minor groove. When you go into a higher level molecular biology course, you will probably have to describe those distances and what the significance of the major groove versus the minor groove is. Largely, it's easier for binding to take place with DNA that is double-stranded in the major groove for the most part. So these strands are anti-parallel to each other, which means that the 3' end of one strand is the 5' end of its paired strand. So these are all associated through hydrogen bonding. Between the T and A, we have 2 hydrogen bonds. Between the G and the C, we have 3 hydrogen bonds. Now this changes the melting temperature, the temperature at which the DNA will separate from each other. Its melting temperature is based on the proportion of GC versus TA or AT. Okay. So again, the hydrogen bonding, well, this is just reinforcing what I just said. But we have 3 bonds between guanine and cytosine, 2 bonds between thymine and adenine when things are working normal. So we denature. So denature equals melting, by the way. When we say we melted the DNA, we're talking about having denatured it, separated it to single-stranded state. And then if you cooled it, you can it'll re-anneal. And this is definitely taken advantage of in the process of PCR, which again, we'll go through that next week. Expect. And to remind myself, why was I talking about vertical gene transfer here? Let's see. I'll leave myself a note. I spent too much time. There we go. Okay. Well, I don't know. Now, Barbara McClintock, I think I mentioned her before. Her work on maize genetics in the 30s through the 1950s resulted in the discovery of transposons. We didn't recognize the significance at the time. Now this is the inset for the Women in Science and how we're kind of crappy about that right now. I'd rather we do this as a conversation. So I remember to come back to this in lecture, or make a separate standalone video to talk about it and the issues around it. But yeah, there's definitely problems because we have to stop telling girls that they aren't good at science or math, because it's not true. Okay, so clinical focus part 2. Let's see, Alex. Alex has the foodborne illness, so that's what his physician suspects. So some of our options that we have seen before, we have an enterotoxigenic Escherichia coli, our good friend cholera, of course, and then the Campylobacter jejuni, various Salmonella strains, or it could be any number of viruses involved. And of course, finally, our protozoan, our eukaryotic pathogenic friends. Oh yeah, Entamoeba histolytica. I skipped that chapter. I'll try to come back to that, but I want to make sure y'all get through the diseases. Well, I mean, we will talk about it in amongst the diseases. That's how we'll do that. Okay, so the physician orders a stool sample to identify possible causative agents and to look for the presence of blood, because certain kinds of these pathogens are associated with the production of bloody stools. Whoops. So neither blood nor cysts. The physician suspects traveler's diarrhea caused by enterotoxigenic Escherichia coli. And so the physician ordered a diagnostic lab test of the stool sample to look for DNA sequences encoding a specific virulence factor of ETEC. The physician instructed Alex to drink lots of fluids to replace what he was losing and discharge him from the hospital. So when that test is conducted, you're going to extract DNA from stool, which we have a lot of really fancy kits that can do that. You don't have to culture, do an intermediate culturing step. You can extract DNA directly and then run a PCR. And they're looking for a gene encoding a very specific virulence factor. So that's a lot better than just looking at 16S and getting a composition of the normal flora, because you're talking about 100,000 plus organisms or bacteria per gram of feces or more. Yeah. So anyway, this type of Escherichia coli, now again, they carry these virulence factors on plasmids, and they will share those with other Escherichia coli just fine. So we have heat labile enterotoxins, heat stable enterotoxins, colonization factor. Both LT and ST cause the excretion of chloride ions from intestinal cells of the intestinal lumen, causing a consequential loss of water, resulting in diarrhea. CF encodes a bacterial protein that aids in allowing the bacterium to adhere to the lining of the small intestine. So why use genetic analysis instead of either isolation of bacteria from the stool sample or direct gram stain of the stool sample alone? I already answered that. There's a whole lot of organisms involved there, and it would take you days and weeks longer than it needs to, than if you can just PCR for a very specific gene encoding these specific toxins. It's a lot easier to do that. Okay, section 3, structure and function of our... I have to make sure I keep an eye on the time. Okay, we're good. Well, I mean, I know we're good because I only just started, but... Oh, I have my phone here. What? Capital 1. Yes, I did. I guess I don't need to answer that now, but... maybe I should. I decided to pay for this broadcasting software because it seems to work okay. I've used it a couple times before. Nick and Quinn had this idea that they just went through this, that they were YouTubers, but they only made a couple of videos. That could be fun. So let's see. Let's text confirm. IRM confirm. Send. Okay. Hopefully I didn't have to all caps that. All right. Sorry. So we're looking at the basic structure and function of RNA. Oh, I got people joining my Discord too, but that's for something else. Okay. Now let's see here. We are looking at the biochemical structure of ribonucleotides, the similarities between RNA and DNA, the functions of 3 main types of RNA used in protein synthesis, explain how RNA can serve as hereditary information. Okay, I'm going to mute for a sec. Oh, right. Yeah, I used that. I quit smoking in November of 2016. So I'm coming up on 2 years as at least not smoking cigarettes. I do that instead with almost no nicotine left, so that's good. Anyway, RNA structure. So deoxyribose is in DNA, and then it's ribose in RNA. So off that 2' carbon, we have a hydroxyl group instead of just a hydrogen. And then the other major difference is that the thymine in DNA is replaced by uracil in RNA. So it's a similar one-ring structure. It just on this carbon over here, we have a methyl and thymine, and that's just replaced with a hydrogen in RNA to be uracil. Now, DNA is typically single-stranded, but because of that hydroxyl on the 2' carbon, RNA is typically single-stranded. They can, well, and do form, I guess you would call it secondary or tertiary. No, don't call it that. Sorry, I don't want to mess you up. You're messing up Goliad. You can form complicated structures with RNA. because RNA can fold upon itself, and it gets stabilized by short areas of complementary base pairing within the molecule, giving it three-dimensional structure. And it does a lot of jobs. And that contributes to why we think the RNA was first, RNA before protein world. It goes to the basically before evolution, we had to have a molecule that was heritable in some way. And there's this PLA scaffolding that we think happened that led to RNA, and then RNA preceded DNA. So RNA being more like you're running programs, your RAM, and the DNA being more like your hard drive storage. Okay, come on. Oh, change. Okay, basic types of RNAs. Shoof. There we go. So in this chapter, we're only talking about 3 types of RNA, but there's easily 20 or, 30 or something. There's a huge list of different kinds of RNAs. But with respect to protein synthesis, well, there's 5 or 6 at least. But we're only talking about these first 3. We have messenger RNA, and this is pursuant to the central dogma of molecular biology. We have the raw transcript. In eukaryotes, anyway, you have a raw transcript that has to get processed. And you include the exons, you leave out the introns. You also have to package it. You have an untranslated region on either end. You put a cap on the end, and then the poly A tail on the other, so that it doesn't get chewed up too fast. It's able to persist a little bit. And then we have our ribosomal RNA. This is the major structural portions of the ribosomes are comprised of RNA. There's a lot of accessory proteins that make this happen too, but the engine itself is made of RNA, ribosomal RNA. And interacting with those ribosomes, at least the codons of the messenger RNA, are the transfer RNAs that carry the amino acids on the end. And three-dimensionally, they have this this kind of structure here. And then when you see it laid out, it's always this little figure. It's this little bump out right here. Now, we also have microRNAs that interact with the untranslated regions of the messenger RNA. We have long non-coding RNAs that can stabilize or inhibit the messenger RNA, and a ton others. I want to try to do that. Why not? Let's see here. Oops. Right here. All right, so messenger, wait. Wait, this is, we're talking about 4 types. What is it called, the other one? Oh, small nuclear RNA, snRNA. That's kind of useful. Let's see. If I do this, can I turn this on? Into infinity, sorry. So here, we're talking about small nuclear RNA as well. And that includes a group of things like microRNA and whatnot. So it's used in RNA processing in eukaryotes. All right. I'll have to figure out how to do these transitions eventually. All right, great. Hey, 17 viewers. Thanks. That's part of the class. Okay, so moving on. Structure and function of RNA. We're just comparing these 3 types for now. Messenger RNAs are short, unstable, single-stranded RNA corresponding to a gene. Just put that down. They function as an intermediary between DNA and protein, and those are messenger RNA. So ribosomal RNA are long, stable RNA molecules. So 60% of the ribosome's mass is the RNA. So this they ensure the proper alignment of the message, the transfer, and the ribosome during protein synthesis, and catalyzes the peptide bond formation between amino acids. So, like I said, the engine that drive protein synthesis is RNA. Transfer RNAs, they are short, they are very stable, they have extensive intramolecular base pairing, they have an amino acid binding site and a messenger RNA binding site, the anti-codon loop. So it carries the correct amino acid to the site of protein synthesis in the ribosome. Again, we have a different chapter where we go through the process, which I think that's not a bad idea. Now the ribosome itself, the generic, with a generalized illustration here, our cartoon, we have the A site where tRNAs enter. Oh wait, sorry, that's showing the exit site. That's the entry site. Usually this is APE, but this is backwards. I like it, yeah. Anyway, the A site is where the transfer RNA enters. And then a reaction is catalyzed here by the RNA that transfers this growing polypeptide chain to the incoming transfer RNA. So that will move over three nucleotides here to the next codon. And then this one will be in the exit site like this one is right now, leaving. But again, we'll go over that in more detail. And show you a really fun video too. Okay, and the transfer RNA, we have when it's charged, that's when it has an amino acid attached to it. And again, it's highly stable. And I didn't. I have to try to wear it. The genetic code shirt, at least when we do that chapter. All right, so section 4, structure and function of cellular genomes. So gene versus genotype, and differentiate genotype from phenotype. Chromosome structure and packaging, describe that. Compare prokaryotic and eukaryotic chromosomes. Explain why extrachromosomal DNA is important in a cell. All right, so the genotype is the full collection of genes that a cell contains. Everything being expressed or not expressed, recessive alleles, whatever. The raw genetic information that's there in the cell. Whereas the phenotype is the observable characteristics that are derived from the expression of the genotype. And another thing to note here, we have some genes that are always on, always expressed. That's constitutive gene expression of housekeeping genes. Housekeeping genes are constitutively expressed, if that works. Okay, so here's an example of differential gene expression when we're talking about prokaryotes. We have 2 plates of Serratia marcescens here, which I think you've had some exposure to. Well, wait. So in context, exposure sounds bad. We've had this in the lab, right? I don't know if you've seen the red pigment. But normally with Serratia marcescens, if you grow them at body temperature at about 37 degrees Celsius, they will not express the red pigment. If you grow them at about room temperature, 25 would be a little warm, but say 20, 22, whatever, room temperature, 28 here. Sorry, it says it right here in the caption. They will express this red pigment. And if you've seen the red scum in, a shower or a bath that hasn't been cleaned in a long time, that's most likely Serratia. And occasionally we have students that come in that will have Serratia on. I think a couple of y'all did. I saw some red on some of those plates of yours. Okay. So, chromosomal organization. In eukaryotes, chromosomes are typically linear, and many are diploid, though there are haploids. Fungi are pretty fond of having haploid stages, as are some plants. Polyploidy is really popular among the plant kingdom because they can do that. They can double their genome and have offspring that do well. In humans, if we increase the chromosome number of chromosome 21, we have Down syndrome. So DNA supercoiling is used to organize and pack all that chromosomal information. The trivia, whatever, about the length of our chromosomes, if you put them end to end and from just one cell and stretch them out, they would cover about 2 meters, about 3 billion bases. So we have topoisomerases are involved in supercoiling, and that's to prevent overwinding. So DNA packaging, let's go back here first. Okay. So DNA binding proteins, histones, they perform wrapping and attachment to scaffolding proteins. And when DNA is in that condition, it's chromatin. And we have a bunch of other modifications that can be made. Some of these are mediated through proteins that are part of the histone complexes. That's acetylation, methylation, sumolation, and others. When we have these modifications can be inherited. So that's the study of epigenetics. So Lamarck got somewhat exonerated centuries after his death, in that you can pass on traits based on things that happened to you during your life, but as far as I know, these are always bad. They're always bad things. If you go through periods of extreme stress, your gametes will be affected in the way the DNAs are modified, and that can be passed on to your offspring, if you choose to have offspring, of course. Eukaryotic chromosome. Okay, so the first level of organization is none. It's just our basic double helix. And sorry for the creaking noise in this chair. We take that helix and we wrap it around a histone protein complex. And then these histone wrapped segments get condensed into chromatin fiber, which is like 30 nanometers in width. And then those wrapped up coils get further wrapped up, and the chromatin gets condensed. And of course, this is only during metaphase, when they're lined up in the metaphase plane, but during cell division, that's when you'll see the chromosomes all condensed like that. There are sections in chromosomes that will remain condensed, basically a lockdown, like our developmental gene, the homeobox genes that define our head and tail and limbs, our bilateral symmetry. Those genes get shut down permanently. And one of the ideas in regeneration is that we need to be able to reactivate some of those genes. This was the chromosome packing video. Will it work in here? Okay, I'm not going to try to do this here because I don't even know if you can hear the narration. But it's really pretty. We can look at it again. Yeah. The chromatin. All right. I don't even know if you can hear this, so I'm going to skip this. It's just so pretty. Here, let's stop this. Okay. Oops. Whoa. It's going crazy. Okay. Now, in prokaryotes, which is more of our primary focus in this course, their chromosomal organization is a lot simpler. Most prokaryotic genomes their chromosomes are circular, typically, and they are typically haploid, except the Deinococcus, I think I mentioned with the extremophiles, have up to 8 copies of their chromosome. They still require supercoiling. They use DNA gyrase to relieve that. They have histone-like proteins The archaea have proteins that are more eukaryote-like than bacteria do. So, speaking to epigenetic changes, Helicobacter pylori, Shigella flexneri can induce these into the host. That can lead to chromatin remodeling, can impact immunity, and in a way that's potentially inheritable too. So, multiple generational impacts from infections with these guys. Okay, so most of y'all probably aren't even taught this anymore. This was something from several years back now, or more. Oh, we are actually in autumn, aren't we? That's cool. We don't talk about junk DNA anymore. Let's see here. Let me move my head. Oops. Junk DNA. Oh, well, that says prokaryotes under there. Okay. Prokaryotes only tend to have about 12% non-coding DNA. That's what we call it now, non-coding. Eukaryotes have upwards of 98% of non-coding DNA. These are typically found in intergenic regions, as pictured here. So this is non-coding. We have something that codes for a start codon here, have a promoter up here, whatever. But you have these stretches of DNA that don't code for anything. They can still serve structural functions, but you can also have your upstream activating sequences will be up here. I'm assuming in orientation here, obviously, and other features there. So, like if you needed to unwind just a little bit from a histone to get access to it, some of these spacers actually facilitate that. So that's just a lot of words to say we don't call it junk DNA anymore. Here we go. So, and we can't forget the mitochondrion. I think I may have mentioned at some point in your graduate studies, someone's going to be like, where do you find 16S ribosomes? And they're going to try to get you to forget about the mitochondria or the chloroplasts and get you focused on the 18S and the eukaryotic nucleus. But no, we still have these guys here. So the genome of the eukaryotic cell consists of the chromosomes housed in the nucleus, but we also have to remember the extrachromosomal DNA in the mitochondria and in the chloroplasts. Now, these are greatly reduced, the genomes of the mitochondrion and probably of the chloroplast too. I remember it better for the mitochondria. They have a number of their genes have been shifted to the nucleus over evolutionary time, and so they can't survive independently. There's still some talk, and there was that one guy whose name I can't remember right now that claimed to have isolated mitochondria. But there's a more recent paper that talks about that. With the right supplementation, you can apparently keep a mitochondria alive outside of a cell for a while. I don't think you can induce reproduction. You can't culture it, but you can keep it alive to do some protocols with for a little while. I was going to try to look at doing something like that in order to investigate the nitrogen fixation that appears in the Eustalogomatus, but I wasn't able to get around to that in time. So anyway, but this is the importance. So again, with the learning objective, this is the importance of the extrachromosomal DNA. All right. Now, when we talk about extrachromosomal DNA in prokaryotes, we're, of course, talking about plasmids. And in this example here, we have this one Bacillus cereus versus Bacillus anthracis. All we have to do is take these couple of plasmids out of the Bacillus anthracis and put them into cereus. Now, suddenly, you can't tell Bacillus cereus, you can't tell it's any different from anthracis. So it's just the maintenance of these two plasmids that makes that carries the virulence factors, which again isn't only the toxin genes; it's other genes to avoid the host. If any genes on these plasmids help it to sporulate or something, you could include those in virulence factors as well. Virulence factors. So again, the species concept really kind of sucks for bacteria. Okay, resolution. Let's see. Alex, is he going to live? I expect he's going to live because it was traveler's diarrhea. So it was positive for, so the PCR result would have been, they found the gene coding for enterotoxin, the heat labile enterotoxin, the heat stable enterotoxin, and colonization factor, confirming ETEC. Alex's symptoms were not resolving quickly, prescribed antibiotics, and the Cipro worked. Likely got the infection from ingesting contaminated food or water. Emerging industrialized countries are still developing sanitation practices that prevent contamination of water with fecal matter. Travelers in such countries should avoid the ingestion of undercooked foods and water that has not been treated, and seafood, vegetables, unpasteurized dairy products. Yeah, so don't eat the ice, or don't even use the ice. So if you're from the area and you have these endemic ETECs, you're probably resistant to them already. There was also something that Dr. Zimmer mentioned before, that even differences in native flora from your own flora could cause some intestinal symptoms. Not on the scale of, having toxins produced that affect you, but just your flora being disturbed by encountering new organisms. Probably should look that up and source that at some point, if I'm going to mention it. Anyway. All right, so now genome size used to be thought to correlate to organism complexity. That's not true. There are large variabilities in the so kilobase. Oh, well, I did that for those of you that, like me, might see KB and think kilobyte, where it's not in a scale of 1,000, where it's 1,024. But anyway, as you can see here, we have some viruses that can rival the size of genomes of some bacteria. Okay, I said all that before, but this, I guess you can say it trends by complexity of organism, viruses up through mammals. But it's not. This is not a hard and fast rule. There are exceptions. There are some animals that have very little non-coding DNA. Okay, that was the end of chapter 10. So I'm going to make this available. I guess I'll make the assignment available, make it due next week, and I reserve the right to take some questions from it. Let's close this out, and let's look at, in brief, that's like 10 minutes brief. Can I go through everything else in 10 minutes? All right, well, let's see. Oh, wait, I have a summary. Let's look at that. This might facilitate this. Here, I need to close that out. Okay. That's very word dense. I don't know. Let's slideshow that up and see what it looks like. Slideshow. Go. There we go. Oh my. So this is chapter 1. No. Well, maybe. Okay, here, let's just look through this. Fermentation stuff. Leeuwenhoek. You'll probably be answering questions about Leeuwenhoek until you die at some point. I don't know. Let's see. Pasteur, Koch. I don't want to go. Properties of light. Okay, probably not a bunch of refraction questions. We didn't use the equations at all, so not really worried about that. The idea that light's reflected, absorbed, transmitted, yeah, of course. Interference, because you need that with the differential interference contrast microscopes, the DIC microscopes. There we go. Interference contrast. So some of the basic microscopy questions, that's definitely stuff that is askable. Oh, if you actually looked at the review questions, I did take some of the questions from the quizzes from the book directly. Okay. No, I'm not. I'm not really. You have the questions available to you to practice or whatever, and I'm going to take the test. 90% of the questions, at least, will be from those questions. I'm not going to ask more than 50 questions, but it might be fewer. I wouldn't say less than 30. I feel like I should at least stretch to that. I might be using the bubble sheets because that'll be a lot faster to grade. And if that plays nice with me, we'll see. I hate umming too and sewing. Well, not SEWing. I just haven't done it in a very long time. Rambling, obviously, I love to do. What are we at? We are at 10 after. I'm probably going to cut you loose here. Instead, if you have any freak out questions, of course, feel free to email me. I don't think this will be super difficult, but you never know. All right, so I think that'll be the end of this lecture portion today. I can tell you more about what happened with my brother I don't know, next week or something. I'll make sure I'm able to talk about it, you know. But that's where we're going this afternoon. As soon as we're done here, you need to clean up and then go pack up and come back to Mayfield for a memorial this evening. Yeah. Anyway, see you on Wednesday. Bye.